you about is the development of new classes of chemotherapeutics that will go into the clinic next year for the treatment of melanoma and neuroblastoma. For a long time, the actin cytoskeleton has been a very desirable target for chemotherapy. The reason is simply because the actin cytoskeleton is involved in cell proliferation, division, and migration. And as you all know, the problem is that if you incapacitate the actin cytoskeleton, the heart stops beating and the diaphragm stops beating. So the trick has been to find a way to target the actin cytoskeleton but not hit the heart or the diaphragm. And that's what I'm going to explain to you today. Inside the cells of your body, all cells have an actin cytoskeleton. Using advanced microscopy, here is a color-coded Z-stack of a neuroblastoma, and what I want you to understand is the cytoplasm is filled with actin filaments. This is a super resolution image of a neuroblastoma cell, and the message I wanted you to take from this image is, again, the cytoplasm is completely filled with actin filaments. These filaments are doing a range of different jobs within the cell. The composition of actin filaments in our body consists of a primary core actin polymer, shown here as a two-stranded polymer winding around each other. In addition, most actin filaments also contain tropomyosin. Tropomyosin is a separate polymer that runs along the length of the actin filament. In your body, there are over 40 isoforms of tropomyosin. If you look at the evolution of all species and the generation of actin filaments, what you notice is that bacteria have 3 different types of actin, and they do 3 very different jobs. Plants have a much larger number of actin isoforms. In the case of the petunia, there are over 100 actins. But when we look at yeast, the fly, or vertebrates, we see there is a very low number of actins with which we build our cytoskeleton. The difference is that neither bacteria nor plants have tropomyosin. And in fact, with increasing complexity in the metazoans, you see increasing numbers of tropomyosins. So what that means is, in the building of our cytoskeleton, we only use 2 actins, but as evolution has progressed, we recruit more and more tropomyosins to diversify the composition of our actin filaments. This summarizes 30 years' work from my group and is incredibly simple. If you take actins and you combine them with a specific tropomyosin, you generate a filament that has a specific function. So if you take the tropomyosin 3.1, you add it to an actin filament, you will recruit myosin motors, you will inhibit actin severing proteins, and you will generate stress fibers. If, on the other hand, you add this tropomyosin 1.12, you encourage the binding of the actin severing protein, ADF, you inhibit the binding of myosin motors and you generate lamellipodia. If you add TPM 1.7, you also encourage cofilin to sever the filaments, you cross-link them with facin and you generate filopodia. Different tropomyosins, different interactions with actin-binding proteins, different functional outcomes for the cell. What we have discovered over the years is that the tropomyosins go to different actin filaments. They largely form homopolymers. They regulate the amount of actin polymer in cells. They regulate the interaction with actin-binding proteins in an isoform-dependent manner. They control myosin interactions. And more recently, we've just discovered they regulate the MAP kinase pathway and the proliferation of tumour cells. Why do you use tropomyosin? Probably because it allows you to make sure that there is fidelity of filament function. If I have the same tropomyosin along the entire length of a filament, it means that the interactions of actin-binding proteins and myosin motors at this part of a filament will be the same that happen at the other end of the filament. The filament has fidelity of function. It does the same thing at each point along the length of the filament. For some years, it's been clear that the composition of actin filaments in cancer cells is quite simple compared to the composition in normal cells. What this slide shows you is a set of neuroblastoma, which is childhood cancer, and melanoma. And this Western blot is just to show you that the tropomyosin TM5-NM1-2 is constitutive across both neuroblastoma and melanoma, whereas these high molecular weight tropomyosins are highly variable from tumour line to tumour line. Work over the last 10 years has demonstrated that the tropomyosin that you see in these tumour cells regulates cell proliferation. It's limiting for the proliferation of mouse embryo fibroblasts. It's limiting for the size of the fat pad, the kidney, and the brain of a mouse. So the amount of this single tropomyosin, by changing its levels, you can change tissue and organ size in a mouse. And as I said, this is always present in tumour cells. It is responsible for regulating ERK-driven cell proliferation. This tropomyosin, or the filaments that contain this tropomyosin, control the interaction between ERK and important 7 and regulate the entry of ERK into the nucleus to drive cell proliferation. And indeed, what we suspect is the reason that all cancer cells retain this specific tropomyosin to make this specific set of actin filaments is because this is a key regulatory role in driving proliferation of all cancer cells, and it's just recently been published. What we know is therefore that at this last step, where ERK interacts with important 7 and enters the nucleus to drive proliferation, this interaction is dependent on this particular population of actin filaments. So it occurred to us some years ago that we should think about developing drugs that attack this population of actin filaments. And the reasons really are, firstly, these actin filaments that contain this tropomyosin are upregulated in transformed cells. They're an integral component of the actin cytoskeleton of not only neuroblastoma and melanoma, but essentially all tumour cells. They're required for tumour cell growth and survival. They're essential for embryonic stem cell viability, and they're required for promotion of cell proliferation. So it seemed to us, if we could make drugs that incapacitate the actin filaments that contain this tropomyosin, this could become a very effective anti-cancer agent. And that's what I'm now going to describe. How do you make a tropomyosin polymer on actin? The way it is done is that the C-terminus of each tropomyosin dimer overlaps with the N-terminus of the next tropomyosin in line along the length of the actin polymer. So what we immediately thought was, could we destroy this interaction? And that's exactly what we have tried to do. Tropomyosin has very weak affinity for an actin polymer. The key is this overlap that allows a tropomyosin to form a continuous polymer. And that leads to a much more stable structure. So what we did was we used drug design, and it's very simple. The principle of this was very simple. We took, using the computer, the model for the C-terminus of tropomyosin, and of the tropomyosin you find in all cancer cells, and we looked for potential pockets where a drug could bind in the C-terminus. So we ran 25,000 different chemicals in a computer against a model of this structure, and we identified candidate molecules that could bind to this C-terminus. We then tested, using protein chemistry, the ability of the candidate drugs to bind to the tropomyosin. This is the first lead compound, which is called TR100, and this is a model of how it sits in the C-terminus of this tropomyosin. Over here, we're just showing you that there are two arms associated with this chemical, and if you remove either of these two arms, you incapacitate the effectiveness of the drug. Over here, what I want to show you, and this is just shown as a blow-up, is if you expose neuroblastoma cells to the drug TR100, you see as early as 2 micromolar that you're destroying the internal organisation of the cytoskeleton. So that was telling us that we had isolated a compound that had the capacity to destroy the organisation of the actin cytoskeleton. How does it work? Well, I'm going to tell you the truth is we thought it would stop the N-terminus and the C-terminus from interacting, and we thought it would just destroy the ability of tropomyosin to bind to actin. That is not what happens, but it was an interesting thought. Okay. What we found, and we'll just start with this panel here. What we're doing is, if you take actin polymer, so a long polymer of actin, you dilute it, what you find is that it falls apart quite rapidly. If you put tropomyosin onto that actin polymer and then dilute it, you get a much decreased rate of depolymerization. What that means is the tropomyosin protects the actin from falling apart. It stabilizes the actin polymer. If you take the drug TR100 and you incubate it with the tropomyosin, and then you allow it to bind to actin, what you see is that in the presence of the drug, the tropomyosin no longer protects the actin filament from falling apart, and that is just quantified here. Surprisingly, If you allow the actin polymer to form and then expose it to the drug actually has no effect on the ability of tropomyosin to protect the filament. So what that was suggesting was that when you pre-incubate the tropomyosin with the drug, the tropomyosin can no longer protect the filament. Now, how is that happening? Here's the surprise. What we found was that if we look at the binding of tropomyosin to actin, this is a standard binding curve showing the cooperativity of the binding. In the presence of the drug, there's no difference. The drug does not prevent the tropomyosin from binding to the filament. It prevents the tropomyosin from protecting the filament, which would suggest that it is this head-to-tail overlap that is not working properly anymore, and you no longer can protect, the filament is no longer protected. What we noticed when we then looked in cells was that if we took mouse embryo fibroblasts or neuroblastoma cells and exposed them to the drug, we find that in the presence of the drug, the amount of filamentous actin is much more substantially decreased in the neuroblastoma compared to normal fibroblasts. So what that suggests is in the presence of the drug, the actin cytoskeleton is becoming unstable, which is a desirable outcome if that's how we're going to attack the actin cytoskeleton of tumour cells. To find out how if the drug has specificity, we then ask the question, does the drug discriminate between the cytoskeleton tropomyosin that's found in tumour cells and the tropomyosin that regulates the contraction of your heart and your diaphragm? And the way to do that experiment was we made cells that were either expressing an M-cherry tagged target tropomyosin or a YFP tagged muscle tropomyosin that regulates the heart and the diaphragm. We then exposed the cells that expressed these two different types of tropomyosin to the drug, and then over 24 hours we made movies and we asked the question, what happens to the structure of the cells? What you see is after about 15 hours, the target tropomyosin, which was originally organised as big cables, now starts to become diffuse and becomes increasingly diffuse. On the other hand, the muscle tropomyosin from the heart, the diaphragm, is completely resistant to the drug. So what that's telling us is the drug knows the difference between the tropomyosin in a tumour cell and the tropomyosin that's present in the heart or the diaphragm. Now Justine did a similar kind of experiment where instead of using all of the muscle tropomyosin, she simply switched the carboxy terminus. And so she made the carboxy terminus of the target tropomyosin. It was now given the carboxy terminus of muscle tropomyosin. And what she found was that when she did that, the construct was completely insensitive to the drug. So what that confirmed was that it's the C terminus of the cancer tropomyosin that's been targeted by the drug. What is the effect on tumour growth? In a series of experiments looking at mouse melanoma, human melanoma and human neuroblastoma, what we found was that the initial drug, TR100, was able to provide inhibition of tumour growth in each of these models. What we also noticed was that in tumours that were exposed to the drug, the amount of the target tropomyosin was also substantially decreased, suggesting that in fact the target tropomyosin was becoming less stable. The big question was, is there any impact on the heart? What we found is that the intraventricular septal distance was unchanged after exposure to the drug, Most importantly, there was no effect on troponin I levels. So what that was indicating is we've got a drug that will attack the actin cytoskeleton of tumour cells, but at the same time it will not hit the function of the heart. Now, if you think about chemotherapy, you think about combinations. So our collaborators in Columbus, Ohio, and in particular, it's Tim Cripe's group, what they did was they took every chemotherapeutic that is used currently to treat childhood cancer and ran a combination with TR100 to ask the question, is there any natural synergy? And the nice result that came out of this is there are only 2 chemotherapeutics that show any synergy with TR100, taxanes and Vinca alkaloids, and both show about 20-fold synergy. And what that means from a practical point of view is that if you, for example here, with neuroblastoma, run increasing doses of paclitaxel, this is the normal curve you see for cell viability. If, however, you add low doses of TR100, and these doses do not have any effect on the viability of neuroblastoma, but when put together with paclitaxel, what you can see is that by increasing the TR100, what you're doing is making the cells more and more sensitive to paclitaxel. You do the same experiment with vincristine, you get exactly the same result. And what that means is that vincristine and paclitaxel are about 20-fold more active in the presence of the anti-tropomyosin compound. So what we concluded from this was that the first anti-tropomyosin drug disassociates the binding of this tropomyosin to actin from its ability to stabilize actin filaments. It increases the amount of G-actin in tumour cells. It acts on the carboxy terminus of the target tropomyosin, and it slows the growth of neuroblastoma and melanoma. But most importantly, it synergizes with antimicrotubule which, from a practical point of view, given that we're about to go into phase 1 trials, means we're now building into the trials the capacity to quickly follow with combined therapy with antimicrotubule agents. Now, what I described to you was the way in which we found the initial anti-tropomyosin agents. What we wanted to do was also find out, could we use a high-content screen in order to isolate new classes of anti-tropomyosin drugs? Because our starting hypothesis was we'd be best to target the C-terminus of the tropomyosin, but it would be better to use an unbiased screen where we look for any kind of compound that can hit this tropomyosin target. So we've recently run a high content screen. I just want to quickly just take you through how we've done it and what has come out of the screen. We've used SKNSH cells, which are a neuroblastoma that has been cultured for some time. The nice thing about these neuroblastoma is they have an excellent actin cytoskeleton that you can visualize easily in a high content screen. What we have done is we're screening 115,000 compound library. That's a very large number of compounds. The scale of the experiment is that you have in each well, the 384 well plate, you have your neuroblastoma cells, and in each well a different compound goes in. And what you do is after 24 hours incubation, you then image what has happened to the actin cytoskeleton. And you run some controls, of which the most important control is the starting anti-tropomyosin agent, TR100. What happens when you run this is that you have your controls, you have 3 anti-actin compounds, lotruncul and TR100 and jasplatinolide, and then you have your unknowns. What you find is that you can take photos of the actin cytoskeleton in each well, and also a photo of using DAPI, of the nuclei. And you take 8 images of cells from every well, and you then do the high content analysis. And the first thing you do is you turn your image of the actin cytoskeleton into a digitized image where you identify each specific bundle of actin filaments, and then you can run a program that will quantify how long the filaments are, how big they are, how thick they are, plus other parameters associated with the organisation of the cell. And then for every well, ask the question, do you have any drugs that behave in a similar manner to TR100? So in mathematical speak, what this means is that we have approximately 90 different variables that are measured in every well. You then map those 90 variables into what's called 90 space, and you generate a map like this where for every individual molecule you have each of these parameters scored. What you're looking for is parameter hits where the map of behaviour that you score is similar to what you see for TR100. So it just sounds like a massive amount of computer analysis. What comes out of it are characteristic types of organisation of actin. So, for example, just to remind you, here are control neuroblastoma cells. This is what TR100 looks like, and we have a number of compounds that give a similar phenotype. This is latrunculin, for example, and then some of these are phenotypes that we've never seen before in terms of the organisation of the actin cytoskeleton. So essentially what we've created is a sort of drug library which targets the actin cytoskeleton in many different ways, but most importantly, out of this is coming candidate molecules that are very similar in their behaviour to our lead anti-tropomyosin drug. So this is, as you can imagine, somewhat laborious. We've so far screened almost 33,000 compounds out of the 115,000. We've identified 521 that impact the organisation of the actin cytoskeleton, and in fact we have 21 different types of impacts. Of these, we've identified 25 compounds that have a very similar phenotype to TR100, but most importantly, of the 25 TR100-like compounds, 6 have been selected for further testing, and they actually have very similar IC50 profiles to TR100. So we think we've now got a drug pipeline that we can use to develop new classes of anti-tropomyosin compounds which can come behind TR100. Now, the final issue I want to raise with you is the ability to identify the function of an anti-cancer agent such as this in a living animal. One of the things...
Carry on. 10 minutes more.
Okay, great. So one of the things clinicians have asked us is, can you show us in a living animal the way in which the drug works to target the actin cytoskeleton? And that is not a trivial thing to do. But what we have done is taken advantage of the work of Roberto Wiegand's lab at NIH to visualise the actin cytoskeleton in a living mammal and to then watch the impact of the drug on the actin cytoskeleton in real time. And it's done. And just, I guess, to remind you, and this was an issue, I think, of some import to our collaborating clinicians. It's not just can you visualise the drug impacting the actin cytoskeleton in living tissue, but can you measure the relationship between drug blood levels and the impact on the actin cytoskeleton? What I'm going to focus on today is how we've established this assay. The system that we're using is not a tumour. The reason it's not a tumour is visualising the actin cytoskeleton in individual tumour cells at this point in time is too difficult. So what we've chosen to do is use the salivary gland. And the reason we chose to use the salivary gland will become immediately obvious as I start to take you through how you can use this system. Now, the salivary gland is composed of acinar cells with very large granules that release their contents into the canaliculus and then through into the mouth as saliva. The nice thing about the use of this system is you can visualize the role of the actin cytoskeleton. And most importantly, what you can do in this system is you can, like that, you can trigger the formation of an actin cytoskeleton before your eyes in a microscope and then follow what happens to that actin cytoskeleton with time. And it's one of the few ways in which you can do this in a living body. Now, work that's previously been done at NIH by Roberta Wiggett's lab has shown that when you stimulate the salivary gland, these large granules now assemble an actin cytoskeleton around the granule. They recruit myosin motors and they then squeeze the contents of the granule to remove the contents into the canaliculus, which then becomes part of the saliva. This is a 90-second process. So what it means is you can take a mammal and you can expose the salivary gland. In the process of 2 minutes, you can watch the assembly and disassembly of actin filaments in a very controlled manner. And for understanding drug action, this is perfect. This is to remind me to tell you that the tropomyosin target, TM5NM1, is localized to the granules as they're fusing to release their contents. And this is a series of movies, and what I'm going to show you here is that under normal circumstances, you can see the granules forming as they fuse and they collapse to release their contents. In a knockout animal that has this gene knocked out, what you see, go back, oops, okay. What I want you to notice is in the knockout, I want you to notice the size of the granules, because the granules in fact become substantially bigger. And then with the drug, they become even more impressively large. So what this becomes a surrogate measure for the action of the drug. Okay. So what you can do is you can now, in a living animal, measure the size of these granules and follow their decrease in size with time. In knockout animals, we see that the initial diameter of the granules has increased, and with the drug, we see the impact is even more effective. So what this is allowing us to do is now measure the impact of the drug on the actin cytoskeleton as the cell is fusing these granules and releasing their contents. You can measure this with time, and what you see is that the knockout is intermediate between the impact of the drug and what you see with wild type cells. And this is just quantified here. So essentially, what this now means is that we can develop drugs that attack the actin cytoskeleton of tumour cells. We can now image the action of these drugs in living tissue against specific populations of actin filaments as they form and follow the impact on the dynamics. From the point of view of tropomyosins, I think what we've learned over the last 30 years is pretty simple. The tropomyosins have been evolved to regulate the dynamics, the organisation, the mechanics, the pool sizes and the function of the actin cytoskeleton. Interestingly enough, what's becoming increasingly clear is function includes not just structure, but it also includes the ability to regulate cell pathways, signalling pathways, and in particular, cell proliferation is clearly a pathway that's regulated by these tropomyosins. I think on top of that, it's now clear that the anti-tropomyosin compounds really open the door to a therapeutic strategy that is taking the actin cytoskeleton from something that you could never imagine targeting in a living body, now we think we're looking at the actin cytoskeleton as a realistic therapeutic target for cancer, but probably also beyond cancer as well. The work has been done by two teams within my lab. All of the work on visualising the role of the actin cytoskeleton in signalling has been led by Galina, Jeff and Bin, and in collaboration with Roni Seeger at Weissman. And the work on developing the drugs was led by Justine Stain. The mechanism of action was discovered by Teresa Bonello. The high content imaging to develop new drugs has been led by Nicole Bryce. And the imaging of the action of the drugs in living animals has been done by Andreas Matadonskas. We collaborated with Nicholas, Vivian, a lot of work on neuroblastoma with Tim Cripe's group, and again the mechanism of action of the drugs was done in collaboration with Sarah Hitchcock. Thanks very much for your attention.
Thank you.